All right, welcome to part three of this week's PowerPoints, the end of chapter three, beginning of chapter four, and this one focuses on membrane transport. Membrane transport is ways we move materials into and out of a cell, and we need to move things into and out of a cell. We need nutrients in, we need raw materials in, we need waste products out, or products that the cell is producing to leave. Now, there's two basic ways this can occur, passively and actively. In passive transport, we don't need to invest energy to do it. In active transport, we need energy in order for us to be able to move something into or out of a cell. And that energy is going to be probably in the form of ATP, adenosine triphosphate. Adenosine triphosphate is an adenine base connected to a ribose sugar connected to three phosphate molecules. Because this is ribose and not deoxyribose, this would be an RNA nucleotide used to build one of the three types of RNA. And because phosphates have a triple negative charge, they repel each other. So it takes a lot of energy to add phosphate to phosphate. And energy is stored in this bond. And the energy that's stored in ATP is different from a lot of other molecules in that when you kick off a phosphate, a lot more energy goes with the phosphate than goes with other molecules. That makes this very efficient at transferring energy. Certainly not all the energy, because the second law of thermodynamics says that when one of these phosphates gets kicked off, some heat energy has to be lost too. But more is passed on with ATP than with other molecules. And so that makes this the main energy currency of the cell. Okay. It says that down here that ATP couples catabolic and anabolic reactions. Well, if we look at the ATP cycle, here's ATP. When it kicks off a phosphate, energy goes with it. And that energy can be used to fuel the anabolic reactions in a cell. So if we're going to build membrane or build an organelle or make a protein or copy the chromosomes, we need phosphate from ATP to do that. But to put the phosphate back on, which we need to recycle the phosphate back or recycle the ATP, we need energy to put that phosphate back on. Right? That energy comes from the nutrients that the cell can break down to release chemical energy stored in their bonds. And so, they estimate you need about a ton of ATP in your 10 trillion cells in a day to survive. Well, you can't eat a ton of anything in a day, right? So that means we're constantly recycling ATP. We're constantly kicking phosphates off to provide the energy for the cells to maintain, grow, and function and reproduce. And we're constantly breaking down nutrients to put phosphate back on ADP to make ATP. In terms of transport, we'll start with passive transport. Probably the simplest form of passive transport is simple diffusion. Diffusion is just molecules spreading from where there's a lot of them to where there's fewer, from high concentration to low. Here they've taken a drop of food coloring and put it in some water. And where the food coloring first hit, it's in a very high concentration. But over time, the molecules of food coloring are bouncing against each other. There's a lot of elasticity in their collisions, and it's forcing them farther and farther apart. Eventually, if we wait long enough, the molecules of dye will be at an equal concentration throughout this sample of water, and it'll all be a uniform color. Okay. Same thing is true if we open a bottle of cologne in a room. If you're close to the cologne, you rapidly start to smell it first because the molecules are bouncing against each other, and some of them are escaping the bottle, and then they're moving farther and farther through the room. Eventually, everybody smells it, and eventually, whatever it is at equilibrium throughout the entire room. And then we open a door, and it starts to diffuse out into the hallway. So simple diffusion is just the movement of molecules or ions or atoms, right, through a liquid or a gas from high concentration to low concentration. Okay, remember our plasma membrane, right, with our phospholipid bilayer, with the proteins, some form channels, which can rapidly let something through. We'll talk about that. But very small things can diffuse. Gases can diffuse through the phospholipid bilayer. Okay, small ions, small atoms can do the same. Okay, this is showing you again. Here we have molecules of high concentration, and they're slowly starting to diffuse through. Eventually, they'll be at equilibrium, where they're still moving through, but now as one moves in, one's moving out, and they're maintaining equilibrium. This doesn't use any energy. It's counting on the energy of the molecules or ions bouncing against each other to cause this to happen. Osmosis is the diffusion of water. Okay? If you have a solution consists of the solvent. Okay, first of all, solution is a homogeneous mixture. So like the air in the room around you right now is a solution. Okay, if you look at it, you probably can't see the various components in it unless you look in a sunbeam and you happen to catch some of the dust motes floating through the air. Most 80% of the air around you right now is nitrogen. Two nitrogen atoms triple bond to each other. Okay, it's inert. You breathe it in and out all day. It doesn't do anything. Okay, 80% of that. There's also water vapor. There's carbon dioxide. There's some other gases present. There's dust. There's bacteria, viruses floating around. All the things dissolved in the nitrogen are solutes. The nitrogen is a solvent. Okay, in the cytoplasm of the cell, water is a solvent. And all of the things dissolved in the water, the amino acids, the monosaccharides, polysaccharides, the organelles, the nucleotides, nutrient molecules that have come in, everything, ions that are there, all those things are solutes dissolved in the water. Water is a solvent. Now, in osmosis, water will move across the cell membrane to equalize the solute to solvent ratio or concentration on both sides of a membrane. So if I have a special beaker with a semi-permeable membrane in the middle, and on one side I put water with not very many solutes, and on the other water with a lot of solutes, what's going to happen is water molecules are going to go to this side to equalize the amount of water, the ratio between water and the solutes. And now this side will rise. Even though gravity is pushing it down, the tonicity of the water, the equilibrium of the water, is greater than the force of gravity because it wants to be at equilibrium. Okay? Well, this same thing happens in your cells. Okay? And there are three different conditions we could put a cell in. The one I want to talk about first is an isotonic solution. In an isotonic solution, the ratio of water to solute inside the cell is equal to that in the solution outside the cell. So water flows equally in both directions across the cell membrane. Cell doesn't change. This is why when you go to a hospital and you need an IV, they don't give you pure water. They give you a 0.9% sodium chloride solution because it mimics the tonicity of your cytoplasm so that your cells don't swell or shrink. In a hypertonic solution, there's a very high solute concentration outside of the cell, much greater than what's in the cytoplasm. This causes water to leave the cell to try to equalize, and the cell shrinks. This is why we put so much salt and sugar and spices in foods we want to preserve. Because if there aren't any bacteria that are in the food, this causes water to leave their cell membrane shrinks away from the cell wall, a process called plasmolysis, and now that bacteria can't metabolize. It can't reproduce, it can't break down the food, it can't produce toxins. So even though it's still there, and it's not dead, it's just not able to metabolize, it can't cause any harm. Okay? This is why we can't drink seawater, because seawater is hypertonic to the cytoplasm of our cells. It'll cause water to leave our system and cause us to dehydrate. A hypotonic solution is the opposite of that. This is when you have very low solute concentration outside the cell. Water's going to flow into the cell. The cell's going to swell. It may burst. Now, a plant cell wants this condition. It wants to have water flowing into the cell, and we'll see that in a minute. In an animal cell, though, because all we have is a cell membrane, it could cause a cell to burst. This is why you don't drink deionized water, because it's going to cause too much water to flood into your cells. Okay, this is showing a red blood corpuscle in the various solutions. Here in the isotonic solution, typically they're pliable, they have a depression in the middle, okay, and this is normal. Water freely flowing in both directions because the solution outside the cell mimics the tonicity of the cytoplasm. In a hypertonic situation, way more solute outside than inside. Water's going to leave the cell. These cells are going to shrink. They're going to crenate and become dehydrated. And if they become too dehydrated, this can really alter your function. This can become life-threatening. In the opposite situation, in a hypotonic solution, water is going to flood in. These cells are going to expand and swell. You can see this one is burst, okay? And that can be damaging. Animal cells prefer isotonic conditions. Okay, plant cells, they want to increase their internal pressure, what we call turgor pressure. And this is the pressure that their cytoplasm is going to exert against their cell wall, their membrane, or anything else that's holding it inside. And so in a plant cell, remember they've got that large central vacuole, they want that full of water. And so they want very much to cause water to flow into their cell. They want this to swell right up because this puts pressure against their cell wall from the inside, and that helps to make the entire plant strong. Right? If you've ever seen a plant that's lost too much water, it gets all wilty. And the leaves droop and the stems droop, and if it doesn't get water pretty soon, it's in big trouble. Okay, plant cells, even if you water them, it might not be enough to make them become turgid. But when they have that really good high tonicity inside the cell with high turgor pressure, now they've got a lot of pressure pushing on the inside out, and that makes the plant stronger. This is how those really, you ever look at those like 10-foot tall ornamental grasses with a really thin stem, and a big wind will come up that's knocking down trees, and those things are just waving in the wind. Part of that is because of the high turgor pressure that exists inside the cell. The other is because of the strength of their cell wall. Another passive process is similar to simple diffusion, but it's called facilitated diffusion. Okay, small things like gases, water, small nonpolar molecules can easily diffuse across the lipid bilayer, but a lot of other molecules and ions can only cross with the help of a transport protein. Okay, but if we don't need ATP to do this, then it's called facilitative or facilitated diffusion. Remember, proteins have a shape, right? So here's a protein in the membrane that has a shape, and I've got some molecule on the outside of the cell that's in a high concentration, very little inside the cell. When this molecule gets close to this membrane protein, it changes the shape of the membrane protein, and when it changes the shape, it moves the material across the membrane. Once this releases into the cytoplasm of the cell, if this is the inside, then it reverts to its normal shape, and now it can help another molecule through. All right, so there are 4 properties of facilitated diffusion. We're moving from high to low concentration. We're crossing a membrane either into or out of a cell. It could go both ways. We need a helper protein, but it's still passive. We're not using any energy to do this. Active transport is like facilitated diffusion, except now we have to use ATP or some other energy molecule to help alter the protein structure. When we move, and now we're going to use special names. When we bring something into the cell, we're going to call it endocytosis. And when we move something out of the cell, we're going to call it exocytosis. Now, you got to be careful. You can't say endocytosis is in the cell and exocytosis is outside the cell, because both of these processes, part of them is on both sides of the membrane. In endocytosis, you start on the inside and you end up on the outside. In exocytosis, you start on the outside and you end up on the inside. Okay? In terms of endocytosis, there are 3 basic types. There's phagocytosis, where what happens is the membrane starts to bend and reach out and wrap around something on the outside, and then this membrane pinches together and forms a vacuole around whatever we just brought into the cell. And now the overall cell membrane is smaller because we took part of it to make this vesicle. Oftentimes we think of phagocytosis as a way to bring solids into the cell. In pinocytosis, we're going to pull in on the membrane and create a vesicle, or an invagination that then can fuse off. And now the cell is smaller because we've used part of the cell membrane to wrap around whatever was trapped in this indentation. Okay. Typically, we think of pinocytosis as bringing in fluids, although solids are also, some fluids are coming in here, some solids are coming in here. In receptor-mediated endocytosis, there are special receptors on part of the cell membrane that when a ligand binds, it causes proteins on the inside of the cell to scaffold and pull in on that, very similar to pinocytosis, except it's mediated by molecules binding to the cell. And then once we get enough bound, we lose part of the cell membrane to form a vesicle. with whatever bound to the cell receptor on the inside of the vesicle. So this is a way to bring things into the cell, specific things that we want the cell to take in. Phagocytosis, again, sometimes phago means to eat, or so cell eating. This is a way some of your white blood cells, your macrophages, your neutrophils, help to get rid of material in your fluids that doesn't belong, like microbes, viruses, cellular debris. Amoebas are also phagocytic. We'll see those when we get into the lab and start working with pond water. Okay, this is showing you an amoeba that's reaching out around a cluster of bacterial cells, phagocytizing them. Eventually, this will come around and meet, form a vesicle. Now that's on the inside. They'll bind it to a lysosome, digest these, and get energy from it. This is very similar to how macrophages that are in circulation in your body work to clear antigens that don't belong. Bacteria, viruses, protozoans, things that don't belong in your system. Okay, receptor-mediated endocytosis. Certainly, this is a way to get things we want in the cell, but this is also a way some viruses enter the cells. Okay, here's an HIV virion, and it's binding to the CD4 protein, or the G2 protein on the surface of a CD4 white blood cell, CD4 T cell. And once it binds, now it can be drawn into the cell, and that's what allows it to infect this cell. There are people who are born without this protein, this G2 protein, and because of that, HIV can't bind to their CD4 helper T cells, and they can be exposed to HIV, but they'll never catch full-blown AIDS because they can't. This virus can't get into the cell it needs to get into in order to replicate itself. Okay, so one mechanism for fighting HIV might be to come up with something that would bind to this receptor protein and block the HIV from being able to bind. Or let's introduce through gene therapy a mutant gene that produces a different form of this T cell that lacks that protein so that the HIV can't bind. Okay, this is the things molecular biologists do to try to solve problems. Exocytosis is kind of the opposite of endocytosis, where we're going to package something that we want to get out of the cell in a vesicle. That vesicle is going to migrate to the cell membrane. It's going to bind and then fuse and then release its contents outside of the cell. And now the cell is slightly bigger because it just picked up the membrane that was part of the vesicle. This is also how cells grow. We can form a membrane in the Golgi. That's a vesicle with nothing in it. It drifts to the cell membrane, fuses, expands, and now the cell is bigger because it's got more membrane. Co-transport is a type of active transport where we're moving two things across the membrane in opposite directions at the same time. And there are several examples of this in the human body. Probably the most well-known and most important is a sodium-potassium pump. Okay, we use ATP to power this, so we can pump sodium out of the cell but get potassium into the cell. So we've got this special protein in the cell membrane, okay, and sodium are these little hexagons, and potassium are these large ovals, and so this protein has a shape, and the sodiums line up and bind, and then along comes ATP, and it binds a phosphate to this, which changes the shape of the protein, and now we release the sodiums to the outside. Okay, in the meantime, while it's open and it's still attached to the phosphate, potassium can come in and attach, and once enough of them attach, it causes this phosphate to be released, the protein reverts to its former shape, and now the potassium's on the inside. So we're moving two things across the membrane using the same protein, needing the help of ATP to provide a phosphate to cause the protein to change its shape when it attaches, and then cause its protein to change its shape back when the phosphate releases. Okay, co-transport. Membrane trafficking is another form of endo and exocytosis, where we can move patches of membrane from the cell surface to either the inside or from the inside to the cell surface when we bring things in and out. Okay, and This is because the phospholipid bilayer can easily fuse to another phospholipid bilayer that gets close enough to it. And that allows us to easily bring things into and out of the cell by having membrane attaching to the cell membrane and then being released from the cell membrane. So that's the end of this week's material. Next week we're going to cover chapter 4, how we generate ATP.